So the nervous system is one of the most fascinating and complex systems that we have in our body. And so I'm going to try to, we have 1 week to do it. So I'm going to try to do some of the kind of important biologic concepts that, if you're going to go on to further study, things that you would need to understand, as well as some of the things that are just really kind of cool and interesting. Right? So I'm deciding what we were going to talk about in the space of just 2 classes. Okay. Oh, it would help if I turned it on. All right, there we go. Okay, so nervous system. So first we're just going to do an overview of kind of structure and function, just kind of really big picture stuff. So the nervous system is one of 2 communication systems that we have within our body. So in terms of how can my pinky finger communicate with my toe, right, if I want to coordinate them to do a little ballet thing, right? How can one part of my body, how can my intestines communicate with my mouth, right? How do different parts of our body communicate and coordinate with each other? We have 2 communication systems in the body. So the nervous system is one of them. And the nervous system is going to use electrochemical signals, and we're going to break that down today and talk about how that works. And it's really fast, right? So when I tell my leg to move, right, it moves right away. Okay, really quick, rapid on, rapid off. Really quick, short term sending of signals or receiving of signals, right? And so this is going to be produced by nervous tissue, kind of no surprise there. The other communication system that we have in our body is the endocrine system, and we're going to talk about that in a couple of weeks. We just have 1 class period for it. And the endocrine system, instead of using electrochemical signals, is going to only use chemical signals using chemicals called hormones. And so hormones are chemical molecules that travel from one part of the body to another, most often through the bloodstream. They are made by parts of the body called glands. So you have a pancreas, which we talked about makes digestive enzymes. Well, your pancreas also makes hormones that serve as chemical messengers from one part of the body to another. Your thyroid gland up here in your neck. You have adrenal glands on the top of your kidneys. And your gonads, which depending on your body type are either testicles or ovaries, or sometimes even a combination of the 2, those also produce hormones. So these are used for one part of the body to tell another part of the body what's going on or what to do. So the endocrine system is slower, but longer lasting. So we have these 2 different communication systems in our body. All right, so the nervous system, when we talk about communication systems, there's kind of 3 aspects of what's happening. The first is sensory information. So this is information that is coming into the system. It's either coming in from the outside environment, and those would be our special senses, sight, taste, touch, smell, hearing, which we'll talk about in detail next week because they're super fun. Right? So that tells us about our outside world, the environment around us. And then we also have kind of internal senses, right? I know if my own, knee is itching. I know if my shoulder hurts. I know if my stomach is gurgling, right? So this is called sensory information. It is coming either from another part of the body or from outside the body, right? This information is being relayed to the system. So a couple of other examples, right? So remember we talked about chemoreceptors when we did the respiratory system. Those are those receptors in the aorta and the carotids that sense pH and send that information up to the brain. And that's what's going to regulate breathing for us. The medulla oblongata. You're like, oh no, I thought I could forget it. No, you have to remember everything, right? Okay. Stretch receptors, right, in your muscles and tendons, right? Lots of things are sending information constantly. So we're receiving information, that's called sensation, okay? Taking information in, and then we process that information. So if I get the smell of cookies, right, that's then going to integrate with my memories of, oh, I've smelled this before, and it ended up tasting delicious, right? I'm going to integrate that. I'm going to interpret it. So the second function of the nervous system is to integrate or process the information, make sense of it, right? You get the raw data in, and then you make sense of it. So did you ever do one of those as part of a hazing or a party game or anything, especially Halloween time, one of those things where they're like, oh, it's a bowl of eyeballs, right? And you don't get to look, and you just put your hand in, and it's peeled grapes, right? But it feels like eyeballs, right? So that's that, taking that raw data, feels like round, slimy things, and then processing it, trying to make sense of it, or interpreting it. So we're going to process or integrate information. And then we can act, right? So then I can send out signals to do something, right? Maybe I'm going to decide, well, I'm going to grab one of those cookies and put it in my mouth, right? Or maybe if I'm like, oh, it feels like eyeballs, maybe I'm going to go, ew, right? So I'm going to send signals back out from the system to parts of my body to do something. And so that's called motor, because it's often going to a muscle that's going to do an action, but not always, but we still call it motor, or efferent. All right, so we have these three functions, excuse me. So we have sensory input, we have integration or processing, and we have motor output, right? So the direction of the information is what's different. Sensory input is coming in to the central nervous system. So I kind of just said it's coming into the system. It's going to come into the central nervous system, which is the brain and the spinal cord. Integration is happening within the brain and spinal cord, or the central nervous system entirely. And then motor output, signals that are going out from the brain and spinal cord to the body, right? So it's all about if it's coming into, processing around within, or coming out from the brain and spinal cord, because those two together are our central nervous system. They're the masterminds. So we divide the nervous system functionally into central nervous system and peripheral nervous system. So the central nervous system here is in purple. So we have the brain, which is inside our noggins, well protected by the skull for the most part, and the spinal cord, which is well protected by our vertebral column for the most part. And then the PNS stands for peripheral nervous system. So the word peripheral means kind of out to the side, right? So it's not central, it's out. So the peripheral nervous system consists primarily of nerves, right? That's how information is going to travel out to or in from all the different parts of our body. So central nervous system is brain and spinal cord. Peripheral nervous system is the system of nerves. And just like our blood vessels that branch out into smaller and smaller so that we supply all the cells and all the tissues, the nerves do the same thing. So they start out big, and they branch out smaller and smaller so that they can go out to all the different parts of our body. All right. So that's the general overview of function and structure. Let's remind ourselves of nervous tissue. We learned this way back in like week 2, I think. All right, so nervous tissue, fortunately, only has 2 main cell types. So you might remember there are the neurons, and these are the cells that are doing the work of the nervous tissue. They are sending and receiving and processing information. And then the little tiny cells that are all around in here, these little tiny purple dots, those little tiny cells are the neuroglia, right? And they're like their staff, right? If the neuron is Oprah, then the neuroglia are their hairstylist and their personal chef and their personal trainer and their accountant and their banker and their housekeeper and their stylist, right? So the neuroglia are going to support and nourish the neurons. There are different types of them that do different things, protecting them from infection, making sure they have nutrients, all different types of things. But we're just going to refer to them as a group as neuroglia. They're the support cells. So the neuron's the cell that's going to do the work. So let's look at the parts of a neuron. So you have your main cell body, right? Kind of the main part of this cell, sometimes also called the soma, S-O-M-A. So we have the main part of the cell, which is the cell body or the soma. And it has these little branch-like parts coming off of it called dendrites. Dendrites their function is to receive information, to receive signals. Okay, think of them like antennae that are out there to receive information and bring it into the neuron cell body or soma. Inside the cell body is where we're going to find the nucleus. It's where we're going to find all the organelles that we've learned about, right? Endoplasmic reticulum, Golgi complex, and lysosomes. All of those things are going to be in the cell body or the soma. And then we have this long, it almost looks like a tail, and this is called the axon. So that long tail is called the axon. That's the part that's actually going to send a message to another cell. And we're going to go through that today. It is covered by an insulating sheath called the myelin sheath, M-Y-E-L-I-N. So the myelin sheath is kind of like that plastic coating around a wire that insulates it, right? So the reason why wires have that is actually two reasons. Not only to prevent people from touching the wire and getting a shock, but it also speeds up conduction along that wire. And the myelin sheath is going to help speed up conduction of a signal along that axon. So a very similar type of function. So the axon has a myelin sheath that acts as an insulator, speeds up the conduction of the signal. The axon is going to end at these little branchy little things that remind me of like those tree frog toes. And those are the axon terminals. So a terminal means an ending. So like a train terminal, railroad terminal is the end of the line. If you have a terminal illness, it is an illness that will end your life. Terminal is the ending. So axon terminals here at the end of the axon. That's going to be a place where some important things happen. So those are the parts of a neuron. So what are nerves? Well, it turns out that those nerves that we talked about, that are the peripheral nervous system, are basically just bundles of axons. So if you think about an axon like a wire that's going to conduct a signal from one place to another, imagine a nerve then is a whole bunch of wires bundled together. Okay? It's a whole bunch of axons just wrapped up together in some connective tissue, usually a little blood vessel to supply them. So a nerve is nothing more than a bundle of axons, sometimes thousands of them. Right? And what's interesting about nerves is there are no neuron cell bodies in them at all. Neuron cell bodies are almost exclusively only found in the central nervous system. But obviously, I can send signals down to my toe. So that neuron that's in my spinal cord that's going to send a signal down to my toe to tap, that axon is almost 3 feet long. Right? Little teeny microscopic axon this long. It's amazing. Right? I have hundreds of these, all going to all the different parts coming out from my spinal cord. So cool. Now remember that myelin sheath that acts like an insulator? That ends up giving nerves a slightly yellowish color. They kind of have a glistening, almost slightly yellow color. And so I'm going to show an image of a nerve from our cadaver lab. And if you don't want to see it, now's when you would close your eyes. Okay? Here we go. So this is the back of somebody's knee. And this is a nerve coming down the back of the thigh. This is the back of the knee, and you can see it splitting into different nerves to then go to the different muscles and different parts of the leg. Pretty cool. All right, safe to look now. Okay. So a nerve is just a bundle of axons. So a lot of times we'll talk about nerve signaling or we'll talk about axon signaling. It's really the same thing. Okay? A signal travels down a nerve if a signal is traveling down one of the axons inside of that nerve. All right. So again, nerves are bundles of axons. Now the new piece of information here is that each axon only sends information one way. So you can be either a sensory neuron, sensory axon that's sending information from the body to the central nervous system, or you could be a motor nerve, motor neuron, motor axon it sends information from the central nervous system out to the body. One or the other. It's like the highway, right? We have separate lanes for eastbound and westbound. Same thing with axons within a nerve. Most nerves are two-way streets with lanes going each way. Okay? Sometimes we have nerves that only do one way. We have a nerve called the lateral femoral cutaneous nerve. Its only job is to provide sensory information for this part of your thigh. Like, what is that? It only sends information one way, that particular nerve. Okay? But most of them have axons that are sending information each way. All right. And of course, as we mentioned before, nerves are going to go out to all different parts of our bodies so that we can send information to and receive information from all the different tissues. So one of the pieces of feedback that I got was on pacing, and that I go too fast for people to take notes. So I want to check in. And so if you've ever been water skiing, you know, if you want them to go faster, you do thumbs up. If you want them to go slower, you do thumbs down. So what I'm going to ask you all to do is put your fists out. If you want me to speed up, give me a thumbs up. If you want me to slow down, give me a thumbs down. If you want me to keep the same pace, do it out to the side. Or just give me a little. Okay. Looks like overall we're going at a good pace. Thank you. All right. So nerves are bundles of axons. Each axon can only send information in one direction. All right. So let's kind of put it into practice for the case. So Ned falls asleep in class. Right? You don't know anybody like Ned, right? I was never Ned. Ha ha. Right? So he falls asleep in class. And then he wakes up and he's like all tingling and numb in part of his hand and his arm. And he can't even move it. Right? So this is like tingling and numbness and pins and needles to the extreme, right? Can't even move it, right? So goes to Student Health Service, of course. We say, you've irritated your radial nerve. Okay, so let's kind of break this down a little bit. Remember, a nerve is a bundle of axons, and that's what sends your sensory or your motor signals. Sensory, sensation, motor, ability to move. They're everywhere. And there's three main nerves of the arm. There's two on this surface. There's the median nerve, which is the one that gets irritated in people who have carpal tunnel syndrome. There's the ulnar nerve, which is the one right here. If you bang your elbow, your funny bone, and you get that tingling, right? That's because you've just banged on your ulnar nerve. And then the third one is the radial nerve. And the radial nerve does this spiral thing behind the bone in your upper arm. So what ends up happening is, if you're like fall asleep like this, it puts a ton of pressure on that radial nerve. Yeah?
So if anybody here is an art student, you need to protect your elbow actually because I have nerve damage because my ulnar nerve has way too much pressure on it while drawing.
Yeah, and it's so yeah, the ulnar nerve is interesting. It's very vulnerable right here. It's very close to the surface. And so actually sitting in chairs with armrests can damage your ulnar nerve. And, you know, anything where you're leaning your elbow on something can cause damage to the ulnar nerve over time. Absolutely, yeah. So in Ned's case, he compressed his radial nerve against this bone here and squished it. So all the things that were trying to send sensation from here or send motor signals to move from up here down there couldn't get through because of, that's another picture of the radial nerve. This has happened to his axons. Right? So if you squish a nerve, you are squishing and compressing those axons. Now, most of the time, if it's not super severe, they will heal. Right? We've all had our foot fall asleep. I hope, probably, right? And it usually goes away in a few moments, right? You know, it recovers just fine. It's right when it's just like banging and like, and then it's fine, right? It heals really quickly. But sometimes, right, if it's prolonged compression over time or if it's really severe, like sometimes happens in a car accident, sometimes that damage can be permanent, right? But in Ned's case, his the axons should heal up quite well, and he'll be fine in a little bit of time. Questions about that? Yeah.
So when you like pinch a nerve, I most, often have heard this like, oh, you pinch a nerve in your neck. Like you either turn it too fast or like some people just wake up with a pinched nerve. Is that just the nerve, the axons compress?
Right. So the question is about if you pinch a nerve, like if you pinch a nerve in your neck, you try and sleep on it funny, or you turn it funny. It's kind of tricky because there can be one of two things going on there. Sometimes you actually truly have compressed or stretched a nerve, and that's why you're having symptoms. Sometimes, especially with the neck, you're actually getting a spasm of the muscles. And that's a little different, but the symptoms can overlap because sometimes when the muscle spasms, the muscle squeezes the nerve and causes compression of the nerve. So very similar. So if you've ever known somebody who had like back problems from a herniated disc or a slipped disc, usually that's because the disc is then pushing on a nerve. And so that can cause pain, it can cause numbness, it can cause an inability to move certain muscles that are normally supplied by those axons. Okay. All right. So now we're going to talk about how those signals actually travel. So the process of nerve conduction, or really signal conduction down an axon, right? Because nerves are just bundles of axons. So how does that actually happen? How does that work? All right. At baseline, we have to be set up and we have to be prepared. And what we have to have ready is something called a resting potential. I don't know who came up with the word. I don't like it because like resting does not seem like, anyway, it just means nothing's happening yet, right? Potential means it can happen. So what is this? Now remember, we said these signals are going to be electrochemical. So this is getting ready for the electrical part of the signal. So what we're going to do is we're going to separate charges, positive from negative. This is exactly what a battery does to create potential energy. You've got a separation of charges. Mother Nature doesn't want that. As soon as you like open things up, things are going to move, which is electricity when they're charged particles. So this would be like a 9-volt battery. You've got your positive charged ions over here, your negative charges over here. You've got your two little terminals on the top of the 9-volt battery. Right? Inside the axon of a neuron, we have positive charges outside of the axon. Now, the axon is part of the cell, so it's covered by a plasma membrane. So we have positive charges outside the plasma membrane and relative negative charges inside the plasma membrane. So we've got a separation of charges. We have positive charges outside the plasma membrane, outside the axon, negative charges inside the axon, inside the cell, on the inside of the plasma membrane. And in fact, you can measure the electrical difference. So this is a little voltmeter, and the difference is minus 70 millivolts. You can actually measure it. It's an actual electrical potential, just like a battery. Now, on a completely different scale, right? I mean, a 9-volt battery, 9 volts versus 70 millivolts, which are millions of volts, right? Thousands of volts, excuse me, thousands of volts. All right, so we're going to separate. We're going to have positive charges outside and negative charges inside. So how do we do that inside of a cell? We do it with sodium ions. So sodium ions, Na+, have a positive charge. So we're going to use pumps to actively push sodium ions with their positive charge outside of the cell, which is going to make the inside of the cell relatively negative because you have all of these positive charges outside of the cell. Now, obviously, if we're trying to push all the sodium outside of the cell and have very little of it inside of the cell, we're going against its concentration gradient, right? Mother Nature doesn't like that. So this is going to be active transport. This takes work. This takes ATP to do and a pump. And in fact, it's estimated. So the question earlier today was, what's metabolism? And so metabolism refers to all of the chemical work that is being done by our body. This job right here of pumping sodium out takes about 2/3 of our total energy expenditure per day. just to do this. Okay? That's how important this is. Our cells are constantly, our neurons and some other cells, which we're going to talk about in 2 weeks, muscle cells, are constantly doing this. They're constantly pumping sodium outside of themselves to establish this resting potential. When I check somebody's blood, their sodium level will be about 140. If I check inside the cell, it's about 8. Okay? Big difference in concentration. It's absolutely imperative that our body maintains that. All right, so I'm going to do active transport, which takes energy in the form of ATP, which is why we have mitochondria, which take glucose and oxygen, make ATP out of it, generate carbon dioxide as a waste product. Okay, so I'm going to have this resting potential, right? So it's like loading a spring. I'm ready to produce an electrical current now. Then I have to decide that I'm going to send a signal. So the neuron cell body, the soma, is kind of the decider in this example. So based on information that it is receiving, it will ultimately reach what's called threshold. Threshold refers to like a doorway into a new place, right? So if you're on the threshold of something, you're about to move over into something new. So the threshold for the neuron is that moment when it decides, you know what? Here's my resting potential that I did all that work to set up. Now I'm going to put it into action, okay? So now I'm going to go ahead and start the process of sending a signal. So I like the dominoes analogy, right? It takes a lot of work to set this up. Doesn't take a lot of work to trigger it, right? Okay. So we're going to reach threshold. Now, how do we actually make things happen? What is the equivalent of the dominoes falling in the case of an axon? Yeah. Yeah, we need to do something. So what we're going to do is we're going to open sodium gates. We're going to say, okay, we've made all that sodium outside the cell. It wants to get in because nature wants things to balance out. We're going to open the sodium gates right here, and sodium is going to flood inside the cell. We're going to start this at the very beginning of the axon, the part of the axon that's right close to the neuron cell body or soma. When those positively charged sodium ions rush across that open gate and flood into the inside of the axon, that movement of charged particles, that is electricity. So what they're seeing here is the current, the voltage that happens as those positive charges move into the inside of the axon. Mm-hmm.
It's very specific, so I don't know if this makes sense. Have you ever had like a light bulb or a night light that's like almost dead, and as you move closer, it gets lighter, but as you move further away, it gets dimmer? Is that something with like the electricity of our nerves?
I have no idea.
I know it's really specific.
I don't know, and I don't know if that's a real thing or if that's just something that you've perceived or something that's happened to you. I don't know. We do have an electromagnetic field around us because of all the electrical things that are happening in our bodies all the time. It's really, really small though. So I don't know that it's enough to be able to be perceived by something we're not touching, for example. Right? So when I want to measure electrical phenomena that are happening in somebody's body, right, I stick electrodes onto their skin. You can measure it, right? I can measure the electrical activity of your heart by putting a little electrode right on your chest wall. That's an EKG. Right? So you can measure this kind of stuff. All right, so the first step is to open those sodium gates right at the beginning of the axon, right close to the cell body or soma. Sodium ions are going to run in as we get that movement of charged particles. That is an electrical current. Okay? It is called depolarization. Where does that word come from? So polarization means to be on the opposite sides, right? Depolarization, I'm letting them rush in. Okay? So you'll see that word used, depolarization of the membrane. We're letting them come across. All right, so that's how I'm going to start the signal. And then I've got to, so that's how I'm going to start it. And then I've got to have it propagate down the rest of the axon. And what happens is these sodium gates are constructed So that when there's an electrical signal that happens, a depolarization that happens right next to them, that triggers the next gate down the line to open up. So then sodium ions rush in again. And that triggers the next gate to open up. So sodium ions rush in. That triggers the next gate. And it goes on and on and on down the length of the axon. So it's just like dominoes. It's just like the wave in a stadium, right, where it propagates down the length of the axon. So here's a little GIF showing you, right, those positive charges move in, and that causes the gates in the next region to open so that then the positive charges can move in. I know it's pretty basic, but it's hard to find good GIFs. Okay. All right, so that is what is happening. Now, you might be like, oh, but Dr. Hall, like as soon as they rush in, so you can start again, they move back out again. You bet your bottom dollar they do. Right? Those pumps are going to get right back on it. Those gates only open for a moment. Because we want to get ready to be able to send another signal again if we have to. Okay? That's called repolarization. Yeah? Yes. So the question is, does that mean all these signals are binary? And the answer is yes. What does that mean by binary? It means that you either send a signal or you don't. It's yes or it's no. It's a 1 or it's a 0. And that is exactly how neurons work. There's no such thing as sending a stronger signal down one individual axon. You either send a signal or you don't. So you might say, well, Dr. Hall, then what's the difference if you're going to lift your clicker versus if you're going to try to lift this chair? Certainly, you need to be sending a stronger signal to the muscle to lift the chair than to lift the clicker. Yes. How do I do that? Yeah.
When your eyes perceive it as being a bigger object, and then your neurons would adapt to that.
So, yep. So by looking at it, right, I can plan. This is going to take more work. I'm going to need to send more of a signal to the muscle. But if each axon can only send a signal of a yes or a no, not relative strength, if I need a stronger signal, what am I going to do?
Oh, did you send, or would you use more neurons?
Exactly. I'm going to use more neurons. I'm going to use more axons, right? To do this, only takes a couple axons to tell some muscle cells to contract. But if I'm going to lift something big and heavy, I'm going to recruit a lot more neurons to send a lot more signals down their axons so that I can get more muscle cells activated.
So I remember reading a thing where it's like the reason why a lot of people actually are electrocuted, as in like they actually die from electrocution, is all of their body, all of their neurons essentially activate and just like people like throw a match or something.
Yes, exactly. So the question is about when people get electrocuted, right? So what happens if you send signals down all of your axons at the same time, right? You get massive muscular contractions, right? So sometimes why you see people just kind of like, they like leap far away. Or sometimes if somebody ends up grabbing onto like a wet cord and they can't let go because those muscles are being signaled to contract. That's why that happens. Yep. Dangerous stuff. We are electrical. And we are binary. We are ones and zeros in our communication signals with our neurons, which really is kind of creepy when you think about like artificial intelligence and stuff, right? That's how computers work. Oh. Okay, good. All right. So this is just another diagram of the same thing. This propagation of this depolarization down the axon is called an action potential. Why did they call that? It's not potential, it's actually happening. I have no idea. I would have called it like an action signal or an action depolarization or something, not an action potential, but they didn't ask me. It's called an action potential. right? And it is this movement of positive charged ions into the axon, moving sequentially down the length of it, creating an electrical signal. All right. Let's do another case. So Nora cuts her finger, and she needs to get stitches. So she comes to Student Health Services, where we do stitches, and we give her some numbing medicine called lidocaine. So we inject some numbing medicine called lidocaine around the wound, and it takes the pain away. She can still sense some touching, but it doesn't hurt anymore. So she says, how does that work? Because she's curious. And we say, oh, because it stabilizes the plasma membrane and prevents depolarization. All right, let's break that down. Okay, so let's see how lidocaine works. Okay, so remember, first we had this resting potential, we had a separation of charges through the active hard work of multiple little pumps using ATP, pushing sodium outside of the cell, outside of the axon. Then we reach threshold and sense an action potential. We let those sodium gates open, so then we get that movement of charges, bam, 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 like dominoes all the way down the length of the axon. That's what normally happens. What does lidocaine do? Well, this would be what happens without lidocaine. Channels open and sodium can move in. Lidocaine blocks the channels. Lidocaine blocks the gates. Lidocaine blocks the sodium gates. So that even if the neuron reaches threshold and wants to tell your brain, ouch, this hurts, it can't. Because we can't depolarize. We can't send an action potential down the axon. Right? We basically functionally cut off your nerve. Thank goodness for that, right? I'm glad I'm a physician now and not back in the day when it was like, here, bite on this leather strap while 6 burly men hold you down. Yeah?
So there's a sensation for people who are amputees where
Yes.
even though the nerve isn't there, but they still feel it.
Mm-hmm.
Why is that happening?
So this phenomenon of phantom pain, so somebody who's been an amputee, for example, they've lost an arm or a leg, but sometimes they can still feel it. Sometimes they feel pain in it. Sometimes they'll feel tickling. Sometimes they'll feel something else. So the question is, where does that come from? First, I will say I'm not entirely sure specifically, but it's got to be coming either from the neuron cell bodies that those axons used to go to that are still there, because remember, they are in the CNS, they're in the spinal cord. So the cell bodies are still there. The parts of the brain that process and receive that information are also still there. So there's lots of reasons why that could happen. Somebody probably knows exactly what it is, and I don't know. I'll look it up though. It's interesting. Okay. So luckily, lidocaine wears off in a couple of hours. So before too long, Nora's sodium channels will be unblocked and back to normal functioning, right? So that's a good thing. We wouldn't want you to be permanently numb. Okay. About how nerves send signals. Okay. So we've traveled all the way down the axon with our action potential. Sodium ions rushing into the inside of the axon, causing depolarization all the way along the way, causing that whole electrical current to move down. And now we end up at those little frog toes, the end, the axon terminal. And we're going to need to then send this signal to another cell, right? That's the whole point, is we're giving this information to somebody, whether it be another neuron inside the central nervous system, whether it be a motor cell, a muscle cell out in the body. Right? So here we are, we send our action potential down the axon to the axon terminals, and now we need to give that signal, communicate it to a receiving cell. But the problem is, there's an actual physical space here. And electricity doesn't move across empty space. So we're not going to be able to use our electric current to get that signal all the way to the next cell. It worked fine to get it from my neuron in my spinal cord all the way down my leg, all the way to the little muscle inside my toe, the muscle cell. I'm sitting there, I'm so close, but now there's a space, and that space is called the synapse. So the synapse is the space between the axon terminal, the end of the axon, and the receiving cell, the cell that I'm going to give my message to. Okay? So this is a problem. So we can't continue with just the electrical signal. It's not going to work. Right? No matter how much I put my finger outside the socket, I'm not going to get that current, right? It requires direct contact most of the time. So I need to use something else. And that something else is going to be a chemical called a neurotransmitter. So we break that word down, neuro meaning of the nervous system, right? Of nerves and the nervous system. Transmitter, to transmit something is to send it across. So the word tells you what it's doing. It's a neurotransmitter. It's what's going to allow us to send this neuron signal across the synapse. So I have these little bundles, little vesicles full of neurotransmitter molecules. When my action potential reaches my axon terminal, that will signal these guys to release the neurotransmitter, which will drift across the synapse and then dock on the receiving cell. I think I have another picture here. Ooh, I forgot. So you've heard about neurotransmitters before. You've probably heard some of their names before. You don't have to write these down. Okay? I just wanted to kind of like take the opportunity to rope in some pre-existing knowledge that you have, right? So acetylcholine, serotonin, epinephrine, norepinephrine, dopamine, GABA, and there's a lot more. These molecules, these chemical molecules are neurotransmitters. Their purpose is to be able to take the signal from the axon terminal across the synapse to another cell. That's why they exist. That's what they do. Okay? So we basically translate that electrical signal now becomes a chemical signal, right? A molecule that we release that can drift across that synapse. Here's a better picture. So we've got our action potential coming down to the axon terminal, right? So the big long axon from the neuron cell body up there. When that reaches, it's going to tell these little vesicles, these little bubbles with neurotransmitter molecules inside to release them into the synapse, into that space in between the two cells.
So I know you said like the neurotransmitters will go on to the next thing in dock, but then do they go back, or is it just strictly one way?
Ooh, good question. So what happens to the neurotransmitters after they get released into the synapse? So a lot of them are going to drift across and dock on these special receptors that the receiving cell has. And when they dock on that receptor, the signal gets transmitted. Okay. Here's another picture. So the reception is really important. You need to have special receptors for the neurotransmitters to dock on. In fact, there is a medical condition where the person has no receptors for a hormone, so it's a little bit different, but I'm just talking about why receptors are important, a hormone called testosterone. And those individuals, even if they are XY, will end up almost entirely female-bodied, even though they have testicles that make testosterone. So if you don't have the receptor, you can't see it. You have to have the receptor. Same goes for neurotransmitters. You have to have a receptor. And the receptors are specific for that neurotransmitter. Serotonin receptors will only allow serotonin to dock and send its signal. Acetylcholine receptors will only allow acetylcholine to dock and transmit its signal. So they're specific to each different neurotransmitter. All right. Now, depending on the neurotransmitter, it may stimulate the receiving cell or suppress it. Right? So depending on the neurotransmitter, it might be giving a different message. Saying do more, do less. Get busy, take a nap. Here's another picture. So we have our axon, or excuse me, our action potential traveling down the axon gets to the axon terminal, where we run into the synapse here. So we're going to release the little vesicles that contain neurotransmitters that move across the synapse and dock on those specific receptor sites that allow them to transmit the signal to the receiving cell. So what happens to those neurotransmitters? Right? We need to turn the signal off. Right? Otherwise, when I picked up my thing right now, it's like, ah. Right? I need that signal to go away. I need it to be transient and temporary. Right? So I don't want to keep telling those muscles. I mean, maybe I do, but I want it to be short-lived. Okay? So there's two different ways I can do that. One is reuptake, and the other is I can use an enzyme. So depending on the neurotransmitter, I'm going to do one of those two things. So we're going to talk about the enzyme first. And the example I'm going to use is the neurotransmitter acetylcholine. Acetylcholine is the neurotransmitter that's used when a motor neuron, so a neuron that's sending information from the CNS out to the body, is giving a signal to a muscle cell to contract. We use acetylcholine for that. Okay? Acetylcholine. So acetylcholine will be released from my nerve, right? When I want to pick something up, it's going to signal my biceps muscle here. It's going to bind to the receptors, and in this case, it would be a receiving muscle cell. It's going to do the job that I'm asking it to do. Now, in order to tell it, okay, let go, and stop telling it to contract, what I'm going to do is I'm going to use an enzyme. So enzymatic means using an enzyme. Degradation means breaking down. To degrade something is to break it down. So I'm going to have an enzyme, in this case called acetylcholinesterase. You have ACE on the end of something, that means it's an enzyme, like lactase, to digest lactose. So I'm going to break down the acetylcholine into its component molecules, thereby rendering it inactive. So the way I think about enzymatic degradation, or breaking it down with an enzyme, is I release that neurotransmitter into the synapse, it binds to its receptor, and then I come around and I chew it up, so it can't work anymore. All right? So if the neurotransmitter is like a cookie, right, the enzyme is like Cookie Monster. Right? I'm just going to chew it up, and then it can no longer send the signal. So this is what we do with our muscles all the time. This is how it works at that synapse between our nerves and the muscles. So that's one way I can end the signal. The other way is reuptake. This is reduce, reuse, and recycle. This is the reuse part, right? So some neurons in their axon terminals, they release the neurotransmitter out into the synapse. It binds to the receptors on the receiving cell, sends its signal, and then, like a vacuum cleaner, I'll suck it back into the axon terminal to use again another time. Serotonin, which is used for a lot of the synapses in our brain, works this way. So these are my little vesicles containing serotonin. My action potential comes down. I release them. They bind to the receptor. And anytime these molecules bind to the receptor, they kind of bind and then they let go and they just kind of drift around. It's pretty casual. They're just kind of hooking up. It's like little one-night stands all over the place. It's pretty cash, right? But then I have these special little pumps that are going to pump the serotonin back into the axon terminal. Reuptake. I'm going to take it back up into the axon terminal. I can use it again later. Right? So that's the other way we can end the signal. So using the cookie analogy, I happen to find what I think is the most adorable GIF ever. All right, let's go to the beginning. He's eating a cookie. Oh, I took it back. Okay? So stealing the neurotransmitter back out of the synapse, back to the axon terminal to use again another day. Okay? All right. I love how the baby's just like, what? Okay. All right. So a couple of cases. So Sam is really interested in understanding sarin, which is a nerve gas. All right. This is a photo of a nerve gas attack that happened in Tokyo in their subway system, actually. Huh? Yes, by a cult. And this is back in 1995, so before your time, but definitely not before. This is actually when I was like your age. No, I was older than you. Oh, that's sad. Okay. All right. But so here we see a victim of the nerve attack, right? So what is sarin? Sarin is of a class of drugs, chemicals, that we call acetylcholinesterase inhibitor. Okay, let's break that down. Acetylcholinesterase, it's got ACE on the end of it, it's an enzyme. Acetylcholinesterase is the enzyme that breaks down acetylcholine. So it's therefore the enzyme that ends the signal to muscle cells to contract. It's an acetylcholinesterase inhibitor. That means it doesn't let the enzyme break down acetylcholine. Therefore, acetylcholine stays in the synapse, keeps sending the signal to the muscle cells to contract. So what happens with an acetylcholinesterase inhibitor? Sarin is one. Many other nerve gases are acetylcholinesterase inhibitors. Many organophosphate pesticides are acetylcholinesterase inhibitors. This is how they work. Is they make it so that all of your muscles, once you send a signal to contract, they can't relax. They stay contracted. Okay? Oops. Oh yeah, this was just a review, right? So resting potential threshold, action potential moves down the axon, reaches the axon terminal. We release the neurotransmitter that goes across to the receptors and then is either broken down by the enzyme or taken back, right? So this is interfering with enzymatic degradation, right? So acetylcholine is what we use in our muscles. So what happens with this acetylcholinesterase inhibitor poisonings, be it sarin, be it some of the other nerve gases, is that your muscles contract and stay contracted. So why does that kill you? David?
So wait, it's like takes out the acetylcholine, which is what makes you stop contracting.
So it takes out the acetylcholinesterase, which is the enzyme that breaks down the chemical signal, that breaks down the neurotransmitter. So the neurotransmitter stays there. Acetylcholine stays there. So what ends up killing you is your diaphragm. Okay? So remember, when your diaphragm contracts down, right, that creates negative pressure in your chest, pulls air in, you inhale. When it relaxes, you exhale. So if you get a big dose of acetylcholinesterase inhibitor, you breathe in, and then you can't breathe out. So you basically suffocate because you cannot move air in and out of your chest cavity. Okay? So it becomes a kind of paralysis in a way because you can't move, but you can't move because the muscles are clenched. So careful around pesticides. All right. Let's talk about Sally. So she's been having troubles of symptoms of depression. And she gets prescribed a medication because part of our understanding of what can cause depression is low levels of serotonin in the brain, which is one of those neurotransmitters that we find in the brain that seems to be important for mood. So fluoxetine, which is Prozac, and a bunch of other medicines like it, are called SSRIs. That stands for selective serotonin reuptake inhibitor. Well, that sounds like a mouthful. Let's break it down. It's an inhibitor, meaning it decreases the ability to do reuptake of serotonin. So let's look at what that would be. So remember, serotonin normally gets released from the axon terminal, and then we have reuptake happening so that then you can release it again. But that's what pulls it out. That's what ends the signal, is that reuptake. So if we block that, then serotonin will spend more time in the synapse, more time sending the signal. So the theoretical construct behind how this class of antidepressants work is that it allows the neurotransmitter, serotonin in this case, to spend more time in the synapse. So it can send its signal. So that's how they work.
Two different questions. So there are multiple SSRIs, like fluoxetine, and then there's also like Zoloft.
Yep.
What are the differences between that? Just like dosage, or how they use them, or
Yeah. So what are the differences between the different SSRIs, right? So there are different ones. So anytime we give somebody medication, we're giving them a specific molecule that's going to act on a process in the body. That's why it's a medication, right? It's going to change how your body functions. So with the SSRIs, they're going to somehow inhibit or interfere with serotonin reuptake. The molecules are similar but slightly different. So some of them might work better in some people and not as well in other people. Some of them might be more likely to cause certain side effects and less likely to cause certain other ones. So it's kind of like different brands of genes. They're all doing the same kind of thing, but there's slight differences.
And then what is the difference between an SSRI and I believe it's an NDRI? Like,
Yeah, so an NDRI is probably a norepinephrine and dopamine reuptake inhibitor, so it's acting on different neurotransmitters. Yep. So we have different medications to treat different types of mood symptoms and disorders, depending on which neurotransmitter we think might be the issue for that person. Yep. So some impact a lot, work on serotonin, some use norepinephrine, some use dopamine, some use GABA. It's amazing. All right. Oh, I blocked my other Re-Uptake. Okay. So just a word about that. So our brains are really complicated. And so this is a real oversimplification, right? But these are some of the neurotransmitters that seem to be most salient or important for mood. I won't test you on this. This is just for your own interest, right? So dopamine we know is really important with motivation, drive, and pleasure and reward. Dopamine is one of the things that gets really stimulated and released in high amounts by heroin and cocaine. Very addictive substances tend to cause a lot of dopamine release. Pornography actually does this too. It's really fascinating. Yeah. Serotonin, right, is really important for mood. Cognitive function, if you've ever taken a serotonin reuptake inhibitor, in the first couple of days as your body's getting used to it, you might have felt kind of like in a little bit of a cognitive fog, like it was hard to think. So we recommend people don't start taking them when they're having an exam coming up, right? But anxiety, irritability, appetite, and sex. So another common side effect of the serotonin reuptake inhibitors is a decrease in libido or sex drive or even sex performance and ability to get and achieve and maintain an erection. Norepinephrine seems to be really important for alertness and concentration, energy, attention, right? But, you know, a little too much of it, you can tip over into anxiety pretty easy with norepinephrine. So this is an oversimplification, right? But these different neurotransmitters seem to be responsible for not only like, making a muscle contract, right? But the kind of those higher order functions in our brain, the things that are more integration and processing that result in thoughts and moods and desires.
So, full disclosure, I'm on SSRIs. I have a friend who just recently started taking them, and she goes, I feel a lot more compulsive now, and I'm looking up there and I see, compulsions and memory and things like that.
Yeah. It's interesting because we'll actually use serotonin reuptake inhibitors to treat obsessions and compulsions, but usually at a really high dose. So it's not an exact science, I'll tell you that. All right. So I want to leave you with a couple more things. I want to remind you, functions of the nervous system. Bring information in, sensory input through nerves, integrate it in the central nervous system, brain and spinal cord, send information and signals out through nerves, that's our motor output. Okay, so we're going to start looking into the structure of the central nervous system now, so the brain and spinal cord. All right. So the central nervous system is pretty interesting to look at. So if I take your brain and I slice it open, it looks like this, okay? And they noticed that it's kind of two-toned, and they were like, well, what's that about? So the outer surface of the brain, for one, it's really wrinkly, but it has this kind of grayish color. And then the more inner, deeper surface of the brain has this kind of whitish color. So they were very creative. They called them gray matter and white matter. So now let's break that down and figure out why do they look different colors. White matter looks white because it is myelinated axons. White matter, what you are seeing is axons, myelinated axons. That's why the inside of the brain looks white. These are all those axons, all those wires going from one neuron to another. The gray part is the actual neuron cell bodies, which is kind of, it makes sense when you think about it, that the neuron cell bodies are kind of on the outside, and then the wires going back and forth, and then the axons are on the inside. It always seems a little weird to me. I'm like, oh, don't you think we'd want to protect those neuron cell bodies a little bit? But no, they're out on the outside. Okay, so when we look at the brain, this is the pattern that we see. Another structure, so besides having gray matter versus white matter, another structural feature of the CNS is that it's covered with membranes. And those membranes are called meninges. So if you've ever heard of meningitis, which can be a really, really serious infection, that's infection of these membranes that cover the brain and the spinal cord. So you bet your bottom dollar it's going to be really serious. It's not often we get an infection of the tissues surrounding our brain and spinal cord. That's why meningitis can be really dangerous. So when we talk about features of the CNS, we have gray matter and white matter. White matter is myelinated axons. Gray matter is neuron cell bodies. We have these membranes called meninges. We also are going to have bones to protect them. So the skull, pretty awesome. Otherwise, I would be like having major problems right now. Okay, so we have this nice bony casing for our brain. For our spinal cord, we need to have more mobility, right? We need to be able to move and do things. So we couldn't have just one big column of bone. So instead we have our vertebral column here, which has a hole running through the middle of it, which is where the spinal cord travels. So both brain and spinal cord are protected by bones. So that's our third feature. The fourth is that within those membranes and within those bones, there's fluid that acts like a cushion and also helps circulate things around. And that's called the CSF, or cerebrospinal fluid. So if I do a spinal tap or a lumbar puncture on someone, because I think perhaps they have meningitis, for example, what I'm doing in that procedure is taking my needle and numbing up their skin with lidocaine first, which blocks the sodium channels. Therefore, they can't send an action potential down their axon, so it doesn't hurt. Do that first. Then put a needle between the bones of the spine into this space around the spinal cord and withdraw some of that cerebrospinal fluid. Okay. So I think we have time for, yes. So I'm going to show you a little bit of a lumbar puncture. The physician who does this is